Hi, I'm going to talk about the AraC proteins, a transcriptional regulator of the arabinose operand in prokaryotes. Among the very popular choices used in genetic circuit design and inducible expression system by bioengineer, often paired with the PBAC promoter. AraC is 292 amino acid long and consists of an arabinose binding domain at the N terminal, a DNA binding domain at the C terminal, and a dimerization domain. Although it is often regarded as an inducible activator, the true story of how AraC regulates this promoter was largely unheard of. In fact, detailed mechanisms elucidated since the 1980s reveal that AraC is both a repressor and an activator, depending on the presence of arabinose. However, such knowledge rarely makes it into mainstream textbooks. AraC can bind to three important sites in the regulatory region downstream of PBAC. In the absence of arabinose, one molecule of AraC binds to I1 and another binds to O2, around 210 base pair downstream. The two proteins dimerize and induce DNA looping, which prevents RNA polymerase from binding to the promoter and start transcribing. In the presence of arabinose, one of the AraC now binds to I2 adjacent to I1, releasing the loop for RNA polymerase to enter. Further inspection revealed that AraC DNA binding domain contains two helix-turn-helix motifs, which interact with the A and B boxes present at all three sites. These boxes are highly conserved, and single substitutional mutation will drastically reduce AraC binding. So how did they uncover the looping mechanism? In one of the key experiments, scientists varied the distance between O2 and I1 by inserting random base pair and testing their repression without arabinose. They found that if the distance was a multiple of 10.5 to 11 base pair, corresponding to a full helix turn, repression was retained at wild type level. But if it was half a turn, repression was hindered due to energetic barrier. And in 2022, my team engineered a minimized PBAC promoter by truncating the I1-O2 distance to only 56 base pair. This retained only 5 full helix turns, but repression was still observed, indicating that looping by AraC dimer is still quite robust to small loop sizes. Interesting question was whether arabinose induction caused conformational changes in the DNA binding domain, such that its binding affinity for O2 is now lower than I2. To test this, the same group replaced I2 with an additional O2 site, and if this hypothesis is true, looping would not occur in the absence of arabinose. However, it was still observed, and if they reverse the directionalities of the distal O2, then looping did not occur. This means that AraC dimer have an inherent preference for looping, not by the difference in binding affinities, but by its geometric constraint. Using these knowledge, recently the Cambridge lab engineered a light-inducible AraC by redesigning the arabinose pocket with a light-sensing domain. The protein, now called BLADE, was optimized to regulate gene expression in response to blue light and serve as an optogenetic tool. Thank you for listening.